again, blank loves this, you have to have the reference genome. You must have the whole genome there from the Human Genome Browser. Aside from that, and we've seen this before, in the Human Genome Browser, remember those back pages? You click on the SNP and it takes you back to that OMIM browser where it basically has all of the total SNPs you could possibly have at every location. That was brought together by the 1000 Genome Project. So a separate thing you should be aware of. You had the Human Genome Browser where you was basically wanted to find that genome. However, not just taking one person's DNA and doing that. I don't know about any of the other SNPs or alleles or VNTRs. So the 1000 Genome Project is basically now combined with the Human Genome Browser because from this information from the 1000 Genome Project, I'm able to identify all of the SNPs everywhere. All of the alleles we see in the population, all of the SNPs we see in the population, and all of the VNTRs we see in the population. And once again, because we're doing it this way, and for a lot of these quantitative traits, we don't just do studies focusing on one gene. We can do this simultaneously for different genes that are all in different haplotype blocks that all play into whatever the disease state is. Once again, simultaneously looking at various different SNPs, part of different blocks that contain different genes within them. In those areas, then, if we see the differences or how things are correlated, where if you have this unique block, therefore that unique allele, will you have the disease state or not the disease state. The one really good thing about this stuff is it's discovery-based, not hypothesis-based. You don't have to be like the null hypothesis, and if not this, then that, and all that stuff. Literally, you just throw all the data on there, and you don't have to say you don't have to think what you're going to find. You do it, and you look at the results. You don't have to worry about making a hypothesis and doing anything that like the scientific method. You can literally you take all the information, or the areas you want to look at, you then compare sequences between disease state, normal population, and then we see what is statistically significant and where do we see differences between those two populations. So it's discovery-based, you don't have to worry about making a hypothesis and then working backwards from there. Once again, all the SNPs associated with, in this case, we're looking at type 1 diabetes. And it's important to note this is on chromosome 6. What's located on chromosome 6? MHC. Why is that important for type 1? Because type 1 is caused by what? Autoimmune disease. MHC is part of that immune system. So on chromosome 6, for type 1 diabetes, we're definitely going to see these haplotype blocks on chromosome 6 are going to be the ones associated with the disease allele and therefore the disease state. Because in this case, we are screwing up MHC, where our MHC, that are being part of all of our other cells, are then targeting the beta cells that they shouldn't be, and therefore then destroying that. Type 2 diabetes, would we be looking at these SNPs in particular for type 2 diabetes? No, because type 2 diabetes has nothing to do with MHC. If MHC is involved and we're destroying our own cells, it's type 1, therefore it's going to be usually this chromosome 6. Type 2, nothing to do with chromosome 6, I believe. Once again, higher up we go, smaller p-value because it's the negative log scale. RS number stands for the SNP. Same thing again. This is the whole lecture. It's just going through this step by step. So being able to get being comfortable with the general flow of how things work, you should be good to go for the exam. So he finally gets the haplotype here on slide 65, but once again, it's the same thing where they're always traveling together. Not always, because you can still have recombination within there. But typically, there's a very low frequency of recombination occurring within that block. So everything in that block always travels with it. And there's not just two haplotype blocks at every block position in the genome. There's a whole host of them because we're talking about population size. So there may be 15 haplotype blocks at this unique area of the genome. And you only have 2 out of those possible 15. But because you have those 2, you make your gametes are always going to have that one or the other one. You're not going to have recombination between the 2. Or if they do, it's not very likely. So that's what they're. MHC and HLA, I'm not going to go into this a lot because this is basically your lecture, the thing to do on your own. I don't remember this that well. You guys are immuno experts. Main thing here, basically you've got the DP gene, DQ, and DR. This stands for recombination, that double thing. So what that means is, once again, what's going on with MHC is it's a random shuffling, right? where basically once the cell goes through the, what is it called? I don't remember. When you like you take one and take the other and put them together.
Exon shuffling?
It's not exon. Well, for this, it's some immuno, whatever it is. What's
The
the term?
hypermutation?
The what?
Somatic hypermutation?
Not a mutation. It's whatever it was from immuno. I don't remember from the first exam. Guess what? I don't remember either. But remember how you have like when the B cells are making the unique antibodies, it's like you take one of these, not one of those, or whatever. I don't. Maybe I'm just confusing everyone. Don't worry about it. But it's whatever that is. But basically here, once you have that going on, after that shuffling's occurred and you've taken one of each, because you have all the different possible scenarios, that cell then when it makes more cells, they only have that unique combination, right? This one plus that one as they go through. One thing to be comfortable with is the idea that within here, for the class 2 in particular, you've got DP, DQ, and DR. The DQ and DR genes, these constitute one haplotype block because there is no recombination within here, or a very low amount of it occurring. Whereas right here, it's a high level of recombination. So therefore, we shouldn't, these cells that are making more cells or going through, when we're talking about making MHC, these DP ones will always, whatever occurred in there will always be the same. But then these, between this and this, I can have shuffling. Those are two different haplotype blocks with the recombination hotspot right in the middle. That's kind of the gist. It's not, make sure you know this fairly well because I don't really understand it that well. Because he goes into detail on like knowing which of these makes the polypeptide, one of these is non-functional. I don't know if he cares about that or not for you guys. So make sure you ask him that today or before he leaves, like before the exam. He's like leaving the last week, right? So make sure you, if you have questions for Blank, make sure you have them together by the end of Thanksgiving because he's gone from all of December. I think that's when your exam is and stuff. So make sure you guys are good with that. But the idea here is that once again, whatever happened in here will always stay together, but these and these are not related. They are not in linkage disequilibrium because there's a recombination hotspot right here. So this combination then will always be together. Within here will be together, but this compared to that, we have no idea. Nothing we can really look at, focus on. The idea of the MHC with that hypervariable region, that's where we're going to have this shuffling going on. We initially are going to make the MHC molecules, and therefore that MHC then will bind, that clef will then bind to whatever it's going to identify, right? And then leading to that immune response. Same thing here, looking at the overall structure of that. This is the last one of the other big things. So once again, haplotypes, and for the MHC, are other things we can look at. It's like any other haplotype block. So if we have a haplotype block, we can then assume which ones go together? DQ and DR, right? Or is it DQ and DP? I don't remember. So so DP, sorry, is together, but then everything that's DQ and DR, those should always be traveling together. So this is one block, and down here, we're going to be looking at this DQ and DR combination. That's what we're doing here. It's a DR-DQ combination because whatever's in that block is always traveling together. Once again, from generation to generation, or parent cell to all the daughter cells, going down on and so forth. So here, just know the nomenclature. The DQ or DR, that stands for the actual gene, and then the star and the number, that stands for the allele. So it's kind of like the SNP. The RS number is the SNP. Here, the actual start with like the lettering, that is your gene, either the DQ or the DR. The asterisk and then the numbers, that is the unique allele of that. Because remember, we all have individual, not all, but we have a wide variety of MHCs, right, between me and you, and that's why we have the issue with organ donors, things like that. So in this case, we know the DQ and the DR, and those are always going to travel together within a haplotype block. And does he say he wants you to even know this? I hope not. I don't know. I would say no, but ask him. And I want to get to the actual important part. Once again, is going to be, is this something that, no, this isn't blank, thank goodness. So, but yeah, once again, they can look at haplotype blocks, and I want to look at, between generations, parent to offspring, certain DR and DQs are going to be traveling together. The DP we're not worried about, because once again, it's in a different area where the recombination hotspot is between them. And the unique thing on here, so I would first know the idea of the autoantibodies to islet antigen 2. And once again, why is this important? What are we talking about here? What's the disease? Let's just look at it here. An autoantibody that attacks our own cells and it's attacking the islet cells. What can we guess? Type 1 diabetes. And we're looking at And some people with this unique haplotype, within there, somewhere in there, aside from that DQ and DR, we're going to have the gene that, with the bad allele, is responsible for that autoantibodies, for the attack on our own islet cells. Same idea as before. So I guess we can use SNPs, we can use VNTRs to identify the haplotype block. We can also use other genes, in this case the DQ and DR genes. We use those as the identifier of the block, and then that'll tell us then, well, if all the disease patients with type 1 diabetes have this haplotype block, something in there then is where we're going to have the issue. Or it could be those genes, it could be the DQ or DR gene themselves. This is what's really important, and it's really confusing the way he puts it up, so I have to draw it out, but I need to keep this on. So, okay, so here basically is what's happening. Basically, so what this slide is telling you is they took from the mom, or I think it's maternal in this case. From the maternal, they looked at the mom, what her haplotypes were for this DQ-DR haplotype, and then they compared it to what the child had. So they're looking at the child's maternal chromosome that came from the mom. They used hers as basically the blueprint for it. So what this tells us is, and because this one should be underlined and the other one should not, to make it make more sense. But here basically what's going on. So from the mom gave us this. DRB1 1501, and then the next one was 0102. I'm going to get rid of the DQ and DRs just to make it easier. And then 0602. It all fits. And then, there's so many. 0103. And the last one is, thank you. All right, can someone read off, what's the bottom one? Can somebody read off to me? Just the numbers is good.
1302, 0102,
Okay.
0609,
Okay.
0103,
Okay.
0601.
Cool. All right, and now can someone read me the top one? The first one, the DRB one, so the
1501,
yeah.
0102, 0609, 0103, 0601.
Okay, cool. So once again,
The top one's supposed to be 0601, not 04.
this one
Yeah.
060 what?
The last one, 04, it should be 06.
Oh, thank you. 0601.
No,
it's
it's
04.
04. Am I reading it wrong?
No, it's
Yeah. So and this is why this is really weird.
So
So
this one is
all right. So here I'm gonna. Okay. So this one right. This is not the same way it's written on here. All right. All right. Here's what's going on. This one right here. This is the child. This is the person of interest. So we're looking at their haplotype. This is this child's maternal haplotype that came from the mom. These two, this is the mom's chromosomes. She had this one, and she had this one. It's chromosome 6. So chromosome 6, chromosome 6. But when you make gametes, what happens? You take these, they come together, recombination occurs, and then you get whatever this is down here. So normally, recombination shouldn't be occurring within this block, but it is. Right? So this is one of the examples where it does occur. Recombination occurred here. How do I know that? I had this and I had this, but I got this. Is this the exact same as either one of these?
No.
No. What happened is that these should be no recombination because they're a unique haplotype block. But what happened is that on the small chance, recombination did occur in here. So what happened is the actual gene that we had, or the actual recombination that we had was this. We started from here, 1501, 0102, 0609. Is that what this one is? No. Guess where that comes into play? Down here, 0609, 0103, 0601. So a recombination event occurred right here. And what happened is that in the mother, when she made her gametes, this got recombined with that. And that led to this unique haplotype. And now this is the haplotype block that the child has. Does this make sense? So once again, I wish this one was one that was underlined, because that would make more sense. That's the child's haplotype block. But it came from these two blocks that the mother had initially. And this is bringing it back to the idea of fluidity. That we are talking on a population scale with haplotype blocks. Those percentages that he writes down on that slide you've seen about 5 times, they're not always the same. Those are slightly changing all the time because recombination events do occur within haplotype blocks, just not very frequently. If we're talking about on a population scale, this will be occurring. So when this occurred, this actually created a new haplotype block. So now, this child has this block, but when they make their gametes, is there a high or low chance this will have recombination within here? Low. It's always low because by definition it's a haplotype block. So this block now should always travel together. Not should, or almost always will, because recombination is not very likely within there. It wasn't likely to occur here, but it did. It can occur even within haplotype blocks. That's the whole point of this. But on the exam, he could give you things like this, and he's going to ask you for, did recombination occur, and if it did, after what point did the recombination, where was the actual crossing over, which would be from here to here, because this is a combination of those two. All right? So, hopefully that makes sense now. I think it's kind of worded weirdly on the article, or at least the way it's presented. Cool. Once again, haplotype blocks going through there. There are 3.1 million SNPs in the human genome, or as a rough estimate. So Blake likes his numbers. So just be so comfortable with that. Key thing for this though is we do not need to sequence and look at every one of those SNPs when we're doing this study. Sometimes we have to. A lot of times we don't though. Because what happens, let's think about it like just statistically. If I've got a disease that only 5 people in the world have the disease, right? Do I want to then, when I'm looking at my data and I want it to be significant, is it better to have more SNPs or less SNPs? More. Why is that? Because if I have a block, can recombination occur in there? Yes. If one of those 5s had a recombination in the block, my data is in really big trouble if I only use 1 or 2 SNPs per block. Why? Because it randomly happened, and even though it shouldn't be that high a percentage, it did. So like 1/5 of my data is basically screwed up. So in that case, I want to have more SNPs that I can identify before I start, because if I have way more SNPs in that block, like all 8 of them, and whatever the example, I'd say I have all 8, then even if a recombination event occurred, I can understand more of that block overall. Let's say on the flip side, I'm looking at type 2 diabetes. So many people have it. If I do it with bigger and bigger numbers, yeah, there's a good chance some people will have a recombination event within a haplotype block, but it's really, really small, so I can use less SNPs to begin with. Because in that case, I don't care if I know data is going to be screwed up, but because I have so much data to work with, so many patients, I don't mind if some of them have recombinations in their blocks. Because even though I'm using a few SNPs, I have so much good data of all the SNPs that are going to be bound with that. So in that case, I don't need to have as much stuff. And that's the idea here, is that the more SNPs I have, if I have smaller data sets, smaller numbers of disease patients, or things where recombination may occur more likely in a block, I need to have more and more SNPs. The only reason this is important goes back to the same thing, unfortunately, it's always about money. If I want to use more SNPs to identify, it costs more money. And the idea is to do really good research, but make it as cheap as possible. Okay? So that's the idea here, is that I want to make sure I have enough SNPs to ensure my data is correct, but I need to outweigh that against the cost of using as many SNPs as possible. Once again, recombination hotspots versus the actual haplotype